Assalamu alaikum class. In this lecture we will solve the remaining MCQs. So come towards the MCQ number 146. The diagram shows some similarities between chloroplast, mitochondria and atypical prokaryotes. So which row is correct? So we have to find out the which particular order of the organelles is correct. So we have read all that options and find out that option number D is correct here. The diagram shows the ultrastructure of a typical animal cell. Which structure synthesizes and transport the lipids? As we all know that Golgi apparatus is involved in the synthesis and the transport of the lipids. So we have to find out which particular structure is the Golgi vesicle. So I can find that option number C is correct here. Some features of the cells are listed: cytoplasm, cell surface membrane, in the ribosomes. Which features are found in both animal and the prokaryotic cells? All the features are present in both animals and the prokaryotic cells. So option number A is correct here. Which size of the ribosome is found in the chloroplast? It is 70S. You simply have to remember it and you can select the correct option. Now come towards MCQ number 150. In which of these organelles is ATP synthesized? ATP is synthesized in mitochondria and chloroplast both. So we have to say that option number D is correct because it overlaps within mitochondria and chloroplast. The electron micrograph shows the part of two cells. This is the boundary, this is one cell and this is the second cell. Which labeled features identify these cells as eukaryotic? W shows that particular double membranous organelle around the cell, and X shows the cell membrane, and Y shows that particular cytoplasm. So we find option number C is the correct here. Plant cells are first screened and viewed using a student microscope. The light source was natural light. What would be clearly visible at 400 times magnification? Using the natural light as a light source, we can only view the neutral light. The chemistry of the mitochondria, grana, chloroplast, and ribosome, they all are too small to be seen with the daylight. Some secretory cells synthesize and release the glycoproteins. What is the correct order of the sequence of the events as they occur in the secretory cells? First of all, the messenger RNA binds to the ribosomes, and then the synthesis of the glycoproteins occurs. Then the product accumulates in the secretory vesicles, and then through the process of exocytosis it is moved out of the cell. So we can find out that option number B is correct here. The scientist mixed the cells with a buffer solution which had some water potential as the cells. He then broke the cells open with a blender to release the organelles. The extracted mixture was filtered and then spun in a centrifuge at a speed to separate the heaviest organelles. This sank to the bottom, forming a solid pellet number 1. This is the pellet 1 and this is the liquid above the pellet. The liquid above the pellet was poured into a clean centrifuge tube and spun in the centrifuge at a higher speed to separate the next heaviest organelle. This organelle sank to the bottom to form a pellet 2. He repeated the procedure twice more to obtain the pellet 3 and the pellet 4, each containing a single organelle. So what is the function of the organelle extracted in the pellet 3? Whether it is the digestion of organelles, it is a function of lysosomes. Lysosomes. Production of ATP is mitochondria. And synthesis of messenger RNA is through the ribosomes. Synthesis of protein is also in the ribosomes. So we have to find out, it can be said to be inside the nucleus. So we have to find out which particular organelle is the higher, comes in the pellet number 3, which is the heavy, so we can say that which occurs in the third number in relation to all these organelles. So when we sort out, we find out that option number A is the correct one. Digestion of organelles. The organelle which we will obtain in the pellet number 3 is involved in the digestion of all organelles. Which organelles are enclosed in a single phospholipid bilayer and they contain the hydrolytic enzymes? It is lysosomes. So option number C is going to be correct. The DNA of the prokaryotes is naked and circular. Which statement describes how the DNA of the eukaryote differs from that of the prokaryotes? It has a nuclear envelope around it and it is a double helix. It has a nuclear envelope around it and it's circular. It has the proteins attached to it and is a double helix. No. It can be somehow correct but not. It has the proteins attached to it and is linear. Yes, it is. So option number D is going to be correct here. What are found in the chloroplast and the mitochondria? DNA, 70S ribosomes, the messenger RNA, all are found in the chloroplast and mitochondria. So option number A is correct here. Where are the cisternae found in the cells? Cisternae are found in the Golgi body, endoplasmic reticulum or mitochondria. They are found in the endoplasmic reticulum and the Golgi body, not in the mitochondria. So option number A is correct here. It is possible for a bacterium to synthesize a eukaryotic protein. This involves the reduction of a eukaryotic gene into the bacterial DNA, which can be translated. Which explains why a bacterial cell can produce a eukaryotic protein but cannot produce a eukaryotic glycoprotein. What is the reason behind this? Bacteria do not have 70S ribosomes. They possess, right? Bacteria do not have a nuclear envelope. The bacteria do not have the Golgi bodies. Bacteria do not have mitochondria. Golgi bodies are involved in the production of the glycoproteins, that is the glycosylation of the proteins. So glycoproteins, Golgi bodies are absent in that of bacteria. So we can say that option number C is correct here. Which structures are found in both typical eukaryotic cells and the typical prokaryotic cells? 70S ribosomes, 80S ribosomes and the circular DNA. All typical cells, they have asked the typical cells. So we will read it carefully and we find out that the 70S ribosomes are found in both cells and the circular DNA. But 80S is found only in the eukaryotic cells, not in the prokaryotes. So the option number B is correct here. An electron micrograph of a cell shows the large quantities of the rough endoplasmic reticulum and many Golgi bodies. What type of the cell is being viewed? Many Golgi bodies. It cannot be a bacterium. It cannot be a guard cell. It can be a lymphocyte. Lymphocyte can have a lot of Golgi bodies because it is involved in the excretion of the useless substances from the cell. So option number C is found to be correct here. Which cell structures can form the vesicles? Cell surface membrane can form the vesicles, endoplasmic reticulum can, Golgi, all can form the vesicles. So option number E is correct. in this case. One of the smallest virus is the polio virus, which has a diameter of about 30 nanometer. In the 2003, the Mimi virus was discovered, which has a diameter approximately 680 nanometer. Pandovirus was discovered and its diameter was 1000 nanometer. It is greater than that of the both Mimi virus and the polio virus, which now correctly matches a cell structure with a virus that has a similar approximate size. Polio virus has a size similar to that of the ribosomes. Mimi virus has a size approximately close to the lysosomes. Pandovirus to that of mitochondria. We can only relate this if we have already known about the sizes of that organelles. So we find out that option number D is correct. Which cell components are present in all the prokaryotic cells? Cell surface membrane is present, yes. Cell wall is present, yes. And endoplasmic reticulum is not present in all the prokaryotic cells, and the flagellum is not present in all. So option number B here is correct. The electron micrograph shows the part of an animal cell. What will be synthesized in the large quantities in the cell? ATP, glucose and RNA. ATP is produced in the bulk quantity. Glucose is not produced. RNA is produced because it is required for the protein synthesis. So option number B is found to be correct here. Mitochondria are thought to have evolved from the prokaryotic cells that were ingested by ancestral cells, which feature have the prokaryotes lost during their evolution into the mitochondria. Cell wall is absent. Circular chromosome is present and endoplasmic reticulum is present. Ribosomes are present. So option number E is correct. Which statement is correct? A virus is composed of protein coat which may surround the RNA or DNA. It is correct. Eukaryotic plant cell walls contain the peptidoglycans. No, they do not contain peptidoglycan. Plasmodesmata and centrioles are found in all plant cells. No, they are found in some cells. Prokaryotic cells have 80S ribosomes. No. So option number A here is the right one. A cell was supplied with the cytosine labeled with radioactive carbon. In which cell structure would radioactivity be first detected? Endoplasmic reticulum, Golgi bodies, nucleus, ribosomes. Firstly, it will be discovered in that particular nucleus because nucleus has a DNA which can incorporate this particular radioactive cytosine and can retain it. The diagram shows some similarities between the chloroplast, mitochondria and the typical prokaryotes. Which row is correct? Number 1 can be the prokaryote. Number 2 can be the circular DNA which is present in the both prokaryotes, and number 3 can be the mitochondria which is present in both the prokaryote and the mitochondria, and number 4 represents the circular DNA which is present in both mitochondria, and number 5 represents that of the chloroplast. So we have to find out that particular option which is correct. Option number C is found to be the correct one. Which statement supports the fact that the mature plant cells can carry out the same role as the lysosomes? A range of the hydrolytic enzymes can be found within the mature plant vacuoles. Yes, it can be. The glycogen found within the vesicles can be hydrolyzed to glucose molecules. No, it is not always happens. It happens in some cases. Single membrane bounded vesicles are formed from the plant Golgi bodies. No. Vesicles formed from the smooth endoplasmic reticulum contain enzymes. They do not always contain enzymes. They can contain the secretory molecules as well. So option number A here is the right answer. The electron micrograph shows which of the labeled organelle is the site of the protein synthesis. As we all know that the ribosomes are the site of the protein synthesis, so we have to find out the particular option which corresponds to the protein synthesis. So A is here is showing some kind of vacuoles, so it is not the site for the protein synthesis. B can be the if I exactly know that, it can be the mitochondria, and the mitochondria, they have their own DNA and the ribosome, so they can be the protein synthesis occur. C stands for that particular Golgi vesicles. Sorry, C stands for, I think it can be the chloroplast, but it is not the site for the protein synthesis. So D here is showing the Golgi vesicles, and it is also not the site of. So B is the most accurate one. So we find out that option number B is correct here. Inside the cell, a damaged mitochondrion can be surrounded and enclosed by a membrane to form a vesicle. What happens after the fusion of a lysosome with the vesicle? ATP production by the mitochondria increases? No. Because the mitochondria is damaged, how it can produce the ATP? It is the wrong one. Enzymes from the lysosomes repair the mitochondria? No. They degrade any organelle, they do not repair it. Hydrolytic enzymes catalyze the breakdown of the mitochondria? Yes, it is. The mitochondria is released from the cell by the exocytosis. So option number C here is the correct one. Which units are the most appropriate to record the diameter of a lymphocyte and a red blood cells? Micron-meter is the most appropriate unit to measure the diameter of the lymphocyte and that of the red blood cells, as we have discussed earlier. So option number D here is correct one. Which structures will be present in a cell that causes the cholera? Cholera is a disease, right? As we all know, it occurs mostly in the cold weathers and the cold seasons. Circular DNA can cause the cholera. Naked DNA can cause, 70S ribosomes can also cause. Because the cholera is a virus, so for the disease to be present, the virus DNA and the ribosomes should be present, so that they can replicate to more to produce more and more viruses. So in this case, the option number E is found out to be correct. Which of the cell organelles will be clearly visible under a high power 400 times magnification power of the light microscope? Lysosomes? No, they are not clearly visible. Endoplasmic reticulum is not visible. Mitochondria, only the chloroplast can be seen. So we find out that option number D here is correct. What are correct locations of the ribosomes in a typical eukaryotic cell? It can be present free in the cytoplasm. Yes. Attached to the outside of the endoplasmic reticulum. Yes. Attached to the inside of the, no. So option number B here is the right one. Bisking tubing is an artificial partially permeable membrane used to demonstrate the diffusion. Glucose molecules can pass through the pores in the membrane, which are approximately 2.5 nanometer in diameter. Which of the following could pass through the pores? Bacteria? No, they are too small. Bacteria can't pass through them. Hemoglobin? No, it cannot pass through them. Ribosomes can. Nothing can pass through them. So we find out whether there is an option of nothing. So option number D is correct. What are found in both chloroplasts and typical prokaryotic cells? 70S ribosomes and the circular DNA, they are found in both typical prokaryotic cells. 70S ribosomes, it is present only, no. So we have to find out that The option which contains both 70S ribosomes and the circular DNA. So option number A here is the correct one. Which statements above both mitochondria and chloroplast are correct? They contain 80S ribosomes. They contain circular DNA molecules. Yes, they produce ATP. So option number D here is the correct one. Which range of the sizes would include the most eukaryotic cells? Most eukaryotic cells fall under the size of micrometer. It can be about 1 micrometer, 10 micrometer to 100 micrometer. So option number C here is the correct one. A scientist carried out an experiment to separate the organelles in an animal cell by mass. The scientist mixed the cells with a buffer solution which had the same water potential as the cells. He then broke the cells open with a blender to release organelles. Then extracted mixture was filtered and then spun in the centrifuge, and a solid pellet one is obtained. This is the pellet one. The liquid above the pellet was poured again, centrifuge, and then the pellet, and the process was repeated, and the pellet 2 and the pellet 3 and 4 were obtained. What is the function of the organelle extracted in the pellet 4? The organelle which is extracted in the very last will be the present in the pellet 4. The digestion of all organelles, it is a function of lysosomes. So this is the lysosomes in the pellet. It is the mitochondria. It is in the nucleus. It is the synthesis of proteins, the ribosomes. So ribosomes are the most less heavy as compared to the other organelles. So option number D here is the correct one. In the pellet number 4, that particular organelle will be present which is the less heavy as compared to the other ones. So ribosomes is less heavy as compared to the other organelles. So we can say ribosomes are present in the pellet 4 and they produce the protein. So option number D here is the right answer. For which cell component would nanometers be the most appropriate unit of the measurement? Cell surface membrane. Yes, it can be chloroplast. They are measured. Chloroplast, mitochondria, nucleolus are measured in the nanometers. They are measured in the micrometers. So option number E here is the right answer. Which organelles are required for the formation of the lysosomes containing the hydrolytic enzymes? Golgi bodies are mostly involved in the production of hydrolytic enzymes, lysosomes. Mitochondria can also sometimes produce it, and the endoplasmic. So all are involved. So we find out that option number B here is the correct one. What are found in both chloroplast and mitochondria? 70S ribosomes, they are found. 70S ribosomes and circular DNA, they are found. Some 80S, no, it is not found. So the option number B here is correct. Both 70S ribosomes and the circular DNA are present in chloroplast and mitochondria.